everyone. So today we will be presenting the paper called Effective Leveraging of Targeted Search Spaces for Improving Peptide Identification in Tandem Mass Spectrometry Based Proteomics. This is a follow-up paper from the same writer. The other paper was published in 2014 from the same writer Avinash, and the paper was called Utility of RNA Sequence and GPMDB, Protein Observation Frequency for Improving the Sensitivity of Protein Identification by Tandem Mass Spectrometry. Some of the workflow and sample preparation methods have been followed from those experiments, including some of the database searches which were retrieved from there. Today, me and Elizabeth will be presenting this paper. At first, I'm going to start off by introducing some of the terms which will be used in this presentation. So first off is GPMDB. It is Global Proteome Machine Database. MySQL is a database dump of GPMDB. Ensemble is a standard protein sequence database. RNA sequence is whole transcriptome shotgun sequencing. DIA is data independent acquisition. APMS is affinity purification mass spectrometry. MSGF it performs peptide identification by scoring MS spectra against peptides derived from a protein sequence database. iProfit is software which is used for integrative analysis of shotgun sequence. Now, introduction of the paper. So basically what was discussed or explained in this paper was how targeted peptide databases can be created based on frequency of identification in the global proteome machine database. In this paper, what the authors are describing is they demonstrate that targeted peptide databases can be easily integrated into existing proteome analysis workflows and also describes a computational strategy which is used for minimizing any loss of peptides identification which arises from potential search space incompleteness in targeted search spaces. This paper also demonstrates the performance of the workflow, which are done using several data sets of varying size and sample complexity. One of the method workflow which was used was there were two different data sets. One was the targeted peptide database and one was the full proteome database, and both the databases were integrated together to run a database search, which led to increased peptide identification, and merge and rescoring of those data further increased peptide identification. The methods which were used in the experiment were first step was data sets which were collected. So the data sets used were K562 cell line lysate, further tested on K562 human cell lysate, and peptide identification was done using MySQL database. The next step was RNA sequencing data which were collected from HeLa cell line retrieved from an Illumina genome analyzer 2, and proteome reference sequence were collected from Ensemble. The second step was MS data analysis pipeline. So the primary database used were MSGF, and search run was done with trypsin as a cleavaging enzyme. The minimum peptide length were 7 amino acid, and further testing were done using tandem search engine. Processing of data were done using iProfit. Targeted peptide sequence database was the third step. Peptide identification frequencies in GPMDB were derived from MySQL, and those were compared to Ensemble version 76. Proteome database was filtered to only retain the trypsin digested peptides containing no more than one cleavage. The fourth step was targeted protein sequence database from RNA sequencing data. The RNA sequencing data were then aligned to Ensemble version 76 human genome using the TopFat and Bowtie, which are also databases which can be accessed online. This was the last step, and now the results and discussions will be explained by Elizabeth.
Okay, now let's start with the result of the study. In this study, they proposed two workflows for improve the peptide identification sensitivity, and this figure shows the basic targeted workflow and make a comparison between the use of the full proteome database and the targeted peptide database. And as we can see in the result, they get to improve the sensitivity of peptide identification. In this figure, we can see the combined search workflow, and in this case, they used both search from the full database and also from the targeted database, and in this case they use the software Hypernet to merge and combine the results. These workflows allows to improve the sensitivity of identification and also reduce the lost peptides due to the incompleteness of the search space. And in this figure, we can clearly observe the difference between the targeted database workflow and the combined search workflow. We can compare both and see that with the combined search they got better result and a high percentage of improvement. Talking about the missed peptides, they got less missed peptide with the combined search workflow. They also use the workflows with different kind of data, as we can see in these graphics. The patterns are similar for the different kind of data, and in all the data set they get a better improvement when using the combined search workflow. For example, in the RNA-seq data, they got, by using the combined search workflow, they got to improve the percentage of peptide identification and reduce the missing peptides and increase the number of new peptides. So, at the moment of selecting the targeted database percentile threshold or limit, they found that the highest improvement percentage is between the 90 and 97 percentile, 97 percentile database for all the different data sets. Okay. Arriving to the conclusion of this paper, this study has demonstrated the utility of using targeted peptide database derived from the global proteome machine database in improving the peptide identification in different kind of tandem MS dataset. In this study, they proposed two workflows: the basic database workflow, search to improve the peptide sensitivity identification, and the combined database workflow that retains this sensitivity but also prevent any loss of peptides due to the incompleteness of search space. Both workflows proposed in this study can be easily integrated into existing proteomic workflows with little or no modification. The Hypernet software can also be used in similar scenarios to merge and combine together different datasets from different search. Talking about the ethical issues in this study, we found that there is not any ethical issue due to, in this study, they don't work with any dinucleotide beam. About if we think that this paper should be published or not, we think yes, because they are providing a useful tool for proteomic research.
Thank you for watching our video. Hope we made the paper easy and simple for you guys to understand. And
If you have any question, we are happy to answer. And thank you. And
Thank you.
that's all. Bye.